feel comfortable participating in the talk through video, that's more than fine. He will be posting the recording afterwards on social media, just for, again, for networking purposes. We will stop the recording at the end for a non-recorded Q&A session, so that you guys can have a little bit more freedom in your interactions with Jake. So without further ado, it's my pleasure and honor to introduce Jake Higgins today. Jake has been a member of the Ingle Group since his arrival at UChicago. And just to highlight kind of a few of his accolades, he has been awarded an NSF fellowship while during his time here at UChicago as a second year, but did not stop at just doing awesome science. He has really been an integral part of the department through both recruiting as a representative for the Graduate Recruitment Initiative team, GRIT, so much so that he won the Joan Shu Departmental Service Award just a few years ago in 2019, and has all along been continuing to do awesome science that I'm sure we're going to hear about today that go far and above my pay grade. But without further ado, I'd like to give the floor to Jake.
All right, thank you so much. That was so nice. I did not expect that. Okay, so let me share my screen. Okay, can you guys see my screen? And you
yes, we're good.
Sweet. Okay. Okay, everyone. Thank you for coming. As Patrick said, my name is Jake. I'm a 5th year in the Ingle Group. Let me go ahead and change this so I can only see me. Okay. So I'm a 5th year in the Ingle Group, and today I'm going to talk about a couple of projects that I've been working on the last couple of years. And so yeah, as Patrick mentioned, I'm going to stop the recording at the end. That would be a good time for sort of deeper questions about this. But along the way, feel free to interject or ask any sort of clarifying questions. So, I'm going to try to get into a lot of technical depth while still trying to make this talk accessible to a broader audience. It's kind of a hard balance to strike, so feel free to ask me to repeat something or any sort of clarifying questions. Okay, so let me start. So first of all, I want to first sort of introduce you to my field, broadly speaking. So I'm an ultrafast spectroscopist, which means I use lasers to study the excited state dynamics of molecules in the condensed phase. Specifically, what my field is the photosynthetic light harvesting field, which is couched within the sort of broader photosynthesis field. So, when people typically think of photosynthesis, what they think of, hold on just one second. Something's going on. Yeah. So when people think of photosynthesis, they typically think of their sort of high school class for photosynthesis, where, hold on, something's, okay, that's better. Okay, they typically think of their, high school photosynthesis where you have this sort of basic equation where you have sunlight, carbon dioxide, water. This undergoes some really complex biochemical process to produce glucose and oxygen. This is an example of oxygenic photosynthesis, but there's actually a wide variety of diversity of different kinds of photosynthetic processes across the evolutionary landscape. I'm not going to talk about really any of those today, any of the biochemistry. What I'm going to talk about is the photophysics that happens before the biochemistry. So photosynthetic light harvesting is the process by which molecules absorb sunlight and transport that excitation energy down to the reaction center. So I have this depicted over here on the left. You have this is a depiction of what happens in green sulfur bacteria, which is the organism that I'll be studying today. They absorb sunlight over here, sort of in what we call the periphery of the entire complex. They transfer their energy down. And if you look at these time constants, you'll see that these energy transfer times are really fast. This is 1 picosecond, or 10 to the minus 12 seconds. Over here is 100 femtoseconds, which is 10 to the minus 13 seconds. And so they transport this excitation across space then that excitation goes and drives charge separation, which is the initial step in that biochemistry in photosynthesis that I talked about earlier. So that's what we're studying here. So the basic sort of mechanistic tool of that is the pigment protein complex. So over here on the left or on the right, I have the Finna-Matthews-Olsen complex, which is the protein that I'm studying. As pigment protein complex suggests, it's a collection of pigments that are wedged together inside of a protein, right? So the protein has a complex amino acid scaffold, which holds the pigments in place. It's ordered to change the energies of the individual pigments, and it's also able to orient them in a way that causes them to sort of dynamically couple to one another. They dynamically couple to produce a basis of excitons, which typically, when we think about this, whenever you have downhill energy transfer of these excitons, it typically moves an excitation across space, which I'll show you a little bit more detail in the next slide. So as an ultrafast spectroscopist, sort of our goal here is to study the dynamical mechanisms that underlie this process. For example, bath-induced fluctuations. We think of the pigments as the system and the protein and all the other stuff as the bath. But the bath is dynamically fluctuating, and there's been a lot of work in the last 15 years to really understand quite literally how the bath can drive the energy transfer dynamics. So sort of a central question here before I delve into the depth of FMO, how has evolution leveraged the quantum properties of its pigments to enhance its energy transfer efficiency and carry out biological functions? So, these are molecules. All of this is molecular. Molecules are quantum mechanical in behavior. And so does it actually take advantage of the quantum mechanical nature, or is that just sort of like a byproduct or just something that's not as important? Could it all be considered classical or something, for example? To go more into FMO, so FMO comes from green sulfur bacteria. This is actually a really, really well-studied pigment protein complex in our field. It's actually a really well-studied pigment protein complex in all the fields in which it's studied. I believe it was maybe the first pigment protein complex to have its crystal structure solved. But it's been studied for a long time. One, because it offers a couple of experimental conveniences that make it sort of a little bit more easy to study than other photosynthetic complexes. And two, it's a relatively simple system. So as I showed you in the previous slide, there are only 8 sites within the FMO complex, only 8 pigments. And so they couple together to produce a basis of 8 partially delocalized excitonic states. The number 8 is very complex, as you'll see in a little bit, but it's nothing compared to some of the complexity that you see in some of these higher organisms, where there are literally hundreds of different pigments that all supposedly play different kinds of complex roles. So FMO is a really good model system to sort of delve deep into some of the dynamics, into some of the really fundamental aspects of dynamics here. So I've shown you here 3 of the 8 excitons. I'll be really studying these 3. And so this kind of shows you a depiction of how the excitons are delocalized across space. So exciton 4 here is fairly delocalized along this part of the complex. He has most of his localization here. Exciton 2 is quite delocalized, as you can see, and exciton 1 is actually mostly localized on site 3 right here toward the bottom, what we call the bottom of the complex. It's actually from exciton 1, which is the lowest energy exciton, that you transfer energy down to the reaction center, which again drives charge separation. Yes, so as I plotted here, here's a couple of amino acids that are actually really important. I'll point at them here. They're cysteine residues, which are just, basically sulfur groups, SH groups. And the cysteine residues have actually been shown to play a pretty strong role in this complex here. Before I go on, I should be noted that they sort of flank exciton 1 or site 3. And so that's an important detail to keep in mind. So it's been shown that these cysteine residues are actually able to quench excitations in certain conditions, in different redox conditions specifically. So here I have some data from a paper from Bob Blankenship's group in 2016. This was 5 years ago. Bob Blankenship is a big photosynthesis person. He's at the University of Washington, St. Louis, or Washington University at St. Louis. And so they studied this FMO complex in different redox conditions and the mutated cysteine residues. And what they found in this paper is that you have a redox-dependent quenching. So on here, this is a time-resolved fluorescence graph. Here's the delay time in nanoseconds, and this is just relative intensity. The inset is the wild type reduced, and you can see it decays, it's pretty present by 5 nanoseconds here. But if you suddenly oxidize this protein, then the decay time speeds way up, which is pretty strong evidence for quenching. So what I should say is this organism lives in anoxygenic environments. It cannot really handle oxygen very well. And so oxygen actually can do damage to the organism. And the way that it does so is that the excitations can actually generate reactive oxygen species, which can then go and really do some damage to the organism. So this is an evolutionary mechanism that this organism has developed in order to quench excitations under oxidizing conditions in order to protect itself. This is the proposed mechanism by which it does so. So you have here, this is in oxidizing conditions. You have this R group, which is a thiol radical. So that cysteine residue is deprotonated and it's just a radical. Whenever you excite, it undergoes a charge transfer process where the charge gets transferred over to the cysteine residue, and then you have charge recombination to get back down to the ground state. Well, in reducing conditions, this doesn't happen. It just stays in the excited state and it stays there for 2 nanoseconds. And then that's plenty of time for the excitation to get transferred. So this is the sort of foundational study for our research. I'm trying to think, did I miss any details here? Yeah, and this is actually a pretty well-known effect. This is called the dithionite effect. It was shown in the 90s, but this paper really fleshes it out in a lot of detail. So yeah, we see this study using time-resolved fluorescence data, but what we want to understand is, do the cysteine residues and do the redox conditions actually affect the ultra-fast dynamics, i.e. the dynamics that happen in hundreds of femtoseconds? So that's kind of our goal here, is to understand how the redox conditions affect ultra-fast energy transport. So I've shown this here. Here's one of the cysteine residues, C333. Here's the other one, C49, and they're really close to these bacterial chlorophyll sites. So they can really affect sort of the side energies here. This is a big polarizable sulfur residue, and so it can exert some sort of differential electric field on the different sites. And so what we did, we collaborated with the Blankenship group. They sent us 8 samples, or they sent us 4, and then we manipulated them to make it into 8. So we have the wild type FMO protein complex. We have singly mutated cysteine residues, C353A and C49A. This A is alanine. So what it literally means is that the sulfur atom is gone. It actually doesn't change the crystal structure. It does change the crystal structure, but it doesn't change the orientations of the pigments or really any of the positions of any of the amino acids. So it's literally just, you can pretend like the sulfur atom is gone. And then the double mutant. And so what we do to create oxidizing conditions is we just expose the protein to the air. And then in order to create reducing conditions, which again protonates that thiol radical, we just add in a little bit of sodium dithionite, which is a strong reducing agent. So that's the setup for everything right now. That's what we're studying. So yeah, again, we're trying to study the ultrafast dynamics. So how do we do that? I'm going to go into now our experimental technique, which is kind of our group's sort of specialty. We're nonlinear spectroscopists, and one of the specific techniques that we use is called two-dimensional electronic spectroscopy, or 2DES. So 2DES is an ultrafast technique which allows us to correlate the excitation energy of a system with the detection energy of a system. We're able to correlate those energies and watch that correlation evolve through time. So what we call this the waiting time, or T2. So here's a little bit of details about what that actually means, or what we're literally doing in the lab. What we do is we generate these laser pulses here, which are really well-controlled laser pulses that are really short in time. They're around 10 femtoseconds full width half max temporally. So what we do experimentally is we generate these pulses, and we can really precisely experimentally control the time delay between the pulses with a lot of precision, with like hundreds of attosecond precision, maybe femtosecond precision. So yeah, we take 4 pulses, we put them incident, or the 4 pulses hit the sample. This first one is just a reference pulse. It's used for some post-processing things. I won't get into the details about this guy, but the first 2 pulses, we call them the pump pulses, and then the third pulse is called the probe pulse. And so what we can do, once the 3 pulses interact with the system, they after those 3 interactions, it produces a signal, which comes out at some later time. And so this signal is dependent on the 3 time delays, the 3 time delays between the pulses, which again, we can control extremely well. We then Fourier transform that signal to get frequency domain in omega 1 and then frequency domain in the 3rd domain. And so this omega 1 is the excitation frequency, this omega 3 is the detection frequency, and we watch how those frequencies evolve with time, the waiting time T2, okay? Maybe it would be more helpful to show you actually a 2D spectrum and what I mean by that. So this little guy down here is a mock 2D spectrum. The x-axis is excitation energy, the y-axis is detection energy, and then this is plotted for a specific waiting time. So we have these diagonal features here. We call these the diagonal features where the excitation and detection are the same. We have these below diagonal features where excitation frequency is greater than the detection frequency. And then we have these above diagonal features where the opposite is true. And so what we can do is if we vary T2, we actually watch this feature down here grow in with time, which tells you that downhill energy transfer is happening because you pump the system in one state. I've shown a simple system here where you just have 2 coupled 2-state systems which share a common ground state. So you pump here at EI, you watch it go you watch it go downhill energy transfer, and then you probe at EJ, right? So for example, what you can do is you can click on specific points in the 2D spectrum and see that signal evolve through the waiting time. And so you see the growth of T2, or you see the growth of the signal during T2, which is a strong indicator, for example, of downhill energy transfer. Briefly, I should mention there's these features above the diagonal. They're called excited state absorption pathways. It's basically whenever you're in an excited state population and then you absorb into a, yes, I see Unisine. And then you absorb into like a biexcitonic state. I'm not going to get into a lot of details there. Yes, Naseem, I see you have a question.
Oops, sorry, I was muted. So the detection time, are you Fourier transforming on like the T3 axis? So if you go back 1 slide.
Yeah.
So is detection time one of the axes that you're, because like I was wondering, like you don't have control over the detection time, do you? Because you don't control when the signal is produced.
Yeah. So that's a good question. That's a good catch. So what we literally do is we take this signal and we bounce it off of a grating, which puts it actually into the wavelength domain, which we then interpolate to frequency, and then we can put it into the time domain by inverse Fourier transforming. So yeah, that's how we actually have control over the detection time. But for simplistic purposes, I just kind of teach it like this. Does that make sense?
Kind of.
Okay, we could talk after. But that's a good question. We don't literally scan over T3, but we have the signal as a function of T3. What we have is the signal as a function of lambda 3. Yeah, but that's a good question. We can talk more. Okay, so downhill energy transfer. Again, we're studying photosynthetic pigment protein complexes and their energy transfer mechanisms and all this stuff. Oh, I should also say before I move on, is that these 2D spectra are actually, they have like a really rich amount of information in them. So, I just showed you one little time trace, but you can see these time traces sometimes wiggle. Those are called coherences, and those are sort of really sensitive probes that you can study the energetic couplings within a system. and you can extrapolate bath dynamics based on how long those wiggles persist. You can also look at line shapes, which tell you a lot about the sort of ensemble behavior of all the molecules in the system, because they're not all behaving the same. But what we see is an average, but you can back out that sort of ensemble information. So anyways, there's a lot of information in a 2D spectrum that one can use to glean aspects about the system dynamics. And so a few years ago, some folks from the Engel group studied the redox-dependent FMO, the redox dependence of FMO. And yeah, I've just shown here the wild type samples. So here you have wild type oxidized at T equals 40 femtoseconds and T equals 2 picoseconds, or 2000 femtoseconds. And what you can see, if you look at these little below diagonal features, you can see that they grow in with time, which again indicates downhill energy transfer. The same thing is true for the wild type reduced. You see downhill energy transfer. And if you plot them on top of each other, they actually saw that the wild type reduced sample grows in with more amplitude. And so, that's an example of, they were able to sort of, this is evidence for the idea that in reducing conditions, downhill energy transfer is more efficient. So that's an example of how you can use 2D spectra to study differences in dynamics between systems. So it's really on this analysis on which I'm basing a lot of my experimental, or a lot of my studies. So they published this paper a couple of years ago, and then I went in, me and some other folks from the group, including Lawson Lloyd, who I think is here. We studied the single mutants, and we studied, we really processed the single mutant data, and they already had the wild type and the double mutant data. And so what I was wanting to do was to look a little bit more in depth into the 2D spectra and see if we can get a little bit more specific information out of these spectra. And so what I did was I developed this analysis method. Okay, full disclosure / warning, the next few slides might be a little bit intense, this is the most mathematical slide in this whole presentation. If you want to zone out for a couple slides, I promise we'll get back to FMO, but I want to talk briefly about spectroscopic theory and this technique that I developed a couple of years ago. But again, please stop me if you have questions, and I can talk more at the end about specifics, because I'm actually going to fly over it. So I'll give you the point from the outset. The point is to extract energy transfer time constants from these spectra. We want to literally know how long does it take for exciton 2 to transfer to exciton 1? How long exactly does it take for exciton 4 to transfer to exciton 1? Because we have an idea of that from the 2D spectra, but all these transfer pathways are extremely congested within themselves. That's the goal. So to go into a little bit of spectroscopic theory, the fundamental observable that we see in our data is this thing called the nonlinear polarization. It's this P term. It's like a semi-classical term where, or at least it's an ensemble term that describes or that is able to produce the signal. So, within this nth order polarization, you have electric fields. These are the laser pulses. And here is this thing called the nth order response function. So in our case, it's a third order response function. So this is the mathematical structure that describes how the system responds in response to the perturbation by the 3 laser pulses. So this is the structure of the nth order response function. You have these little brackets here, which indicate averaging over all of the ensemble space. You have these mu's here, which are dipole operators. And over here, you have the molecular density matrix, which that's the mathematical quantity that contains, your molecular wave functions and stuff like that. And so what you have is a bunch of dipole operators, which are, within a bunch of nested commutators here. And so whenever you expand this, you actually have like a lot of terms here. But, if you take this to its mathematical, logical conclusion, this thing describes exactly how the system is supposed to respond. And what you can hypothetically do is know everything about the system from this. But if you expand all these commutators in the third order response function, you have 8 terms or 2 pairs of 2 pairs of 2 terms, which are, or 2 sets of terms, which are complex conjugates of one another. Whenever you take the dipole operator and its eigenvalues and project it onto the molecular eigenstates, you get more terms. And then whenever you average over all of the ensemble dynamics, you get more terms. So this thing is a beast. And so what we have to do is turn to other tools in order to get a little bit of a conceptual or intuitive understanding of what's going on. And so this is what people do. There's this technique that was, or there's this thing called double-sided Feynman pathways. So this is how to represent, this third order polarization, or this third order perturbation theory. These Feynman pathways, I'm totally not going to go into all the details here. But basically, these are little diagrams. This is a Feynman pathway here in the middle. These are little diagrams that show one particular component of the entire response function. So it's one of these terms here. Yeah. So there's a lot of components to it. These little arrows are the laser pulses that are perturbing the system. These are the time delays. And what I am trying to focus on here is what happens during T2. So during this Feynman pathway that I have depicted here, you see during T2, you see downhill energy transfer from I to J, kind of like this thing I showed you before. And that downhill energy transfer happens with a specific time constant. So what what is postulated from this is each Feynman pathway contributes a small component to the total signal, okay? So for example, this Feynman pathway, the downhill energy transfer during T2 increases, that causes the signal to increase with time. And this happens at a specific point in the 2D spectrum. I won't get into a lot of details. I'll also say that the amplitude is governed by the transition dipole strength. We can talk more about that if you want. But so there's a bunch of different Feynman pathways. They all, if you get all of them, they describe the total response of the system. The only caveat is, yes, you have to get all of them. And they each contribute different relative time-dependent amplitudes to the total signal. So from that, here's what you can do to isolate the time constants. So step 1, you average over regions of the 2D spectrum. So you take a 2D spectrum, you average over here, and what that does is it takes out some of these Feynman pathways, which are due to slow, nuclear or bath dynamics, such as reorganization, reorganization of excited state vibrational modes, or line broadening, spectral diffusion, stuff like that. So that's step 1. Basically, we're reducing the number of Feynman pathways that are contributing to our signal. Step 2, ignore excited state absorption pathways. There's a couple of rationalizations for that, which I can get into if you want, but we're just gonna ignore, remember those negative signals that tend to show up above the diagonal in our kinds of systems. We're ignoring those. We're saying that they contribute relatively less to the signal, okay? And so once you do that, what you're left with is just a few Feynman pathways. My cat is using the litter box. You're left with a few Feynman pathways. These are three different types of Feynman pathways that are shown on the I diagonal and the below diagonal cross peak. This is ground state bleach. You'll see these two things are the same. You see their time evolution over here on the right. Here's a bleach recovery signal, which looks like this little green signal right here. And you'll see the purple and the green have what I'm calling a dynamical symmetry. So in other words, they evolve the same way during T2. Now, if you look at the little orange guys, which are the stimulated emission pathways, which show, those are the pathways that contribute, or that show the excited state dynamics in the system. They evolve differently on the I diagonal and the IJ cross peak. They have what I'm calling a dynamical asymmetry. And so what you can do is you can normalize the I diagonal signal so that it, and then you can normalize the IJ cross peak signal so that they grow with the same amplitude. And then you subtract them from one another. And what that does is it approximately subtracts out the dynamically symmetric pathways. And what you're left with is the orange guys. And so what you can then do is fit to that signal and extrapolate with some fancy techniques, the energy transfer time constants. Okay, that was probably a lot. I'm going to ask if there's any sort of clarifying questions that I can talk about at this point. If not, I promise I will move on to FMO in a moment. Okay, so this is a new technique which you can use to extrapolate the excited state dynamics. So I was a little bit insecure about this for a hot second because I was like, what if it's made up, or what if it's wrong? What if it's not accurate? What if our approximations are not true, or are not, tractable in most systems, or even in our system? And so what I did was I teamed up with a couple of undergrads. So on the left is Chidera Ndife. She's a 4th year chemistry major in the college. On the right, Anna Dardia. She's a 3rd year physics major in the college. So last year, Chidera and Anna both learned this theory of nonlinear spectroscopy, and they spent last summer sitting in their apartments simulating 2D spectra, and the 3 of us were able to sort of numerically study some of the approximate regimes in which this analysis method is accurate. You'll have to trust me, I'm not showing any data because we haven't published the manuscript yet, but for our types of systems, the method is accurate within about 10 or 15%. So we're able to get the time constant information out of those, out of the spectra. I'm super happy to talk to anybody about this if you want to ask me any questions later on, or you can ask them. Okay, so that was it. That was all the spectroscopic theory. Now let's get into some results of the actual spectroscopic data. So taking a turn back to FMO, I'm showing exciton 4, exciton 2, and exciton 1 here. So what we're worried about, or what we're going to fixate on here, is the different excitonic pathways. Different excitonic pathways. So if you're sitting in exciton 4 right here, you're in the excited state, you can either go down to exciton 2 or you can go down to exciton 1. And what we want to know is, in different redox conditions, which one does it prefer, I guess. So yeah, so here's the data. Again, this is the empirical data taken from the time constant extraction method. We have on the left here wild type, the reduced samples, the wild type, the single mutants, the double mutant, and then the same for the oxidized samples. I'm going to take a drink. So this is a bunch of time constant data. Most of these processes happen in hundreds of femtoseconds. Don't look at all the time constants. Let me just point out a couple of trends here. And most of the interesting trends are in the tau 4-1 time constants. So trend number 1, the wild type proteins. Whenever you're in reducing conditions, tau 4-1 happens in 500 femtoseconds. Energy transfer happens in 500 femtoseconds. Whenever you oxidize that protein, which again causes those cysteine residues to become thiol radicals, tau 4-1 suddenly slows down by a factor of 3. Okay. Trend number 2. One thing you'll notice is that there is a little bit of cooperativity in these data. And what I mean by that is, take a look at the reducing data. If you start out wild type, you mutate one of them, suddenly it slows way down, which is kind of strange. Okay, so whenever you take the wild type and you mutate the other one, it doesn't really do anything. But when you mutate both of them, it also slows way down, which makes logical sense. If you do one thing and it changes a lot, if you do another thing, it changes very little. When you do both, it does the same thing as the first one where it changes a lot. So, okay, cool. That's a little bit of a nice little confirmation that our data is okay. But when you look at the oxidized samples for example, tau 4-1 starts off with 1500 femtoseconds. You mutate one, it speeds up. You mutate the other, it speeds up even more. So it stands to reason that if you mutate both of them, it should speed way up. But that's not what happens. It actually slows back down again. So this is a really weird thing. This set off some alarm bells when we first saw this data. But keep that in mind, because we will come back to that, because we will ultimately explain this trend. So, okay. So this is major result number one of this study, is that we see redox-dependent steering of the excitons as a photoprotective mechanism. So as I mentioned before, we're looking at the branching ratios where if you're sitting in exciton 1, are you going to prefer to go down to, or if you're sitting in exciton 4, are you going to prefer to go down to exciton 1, or are you going to go through exciton 2 like this? So in the wild type, based on the time constants, when you're a wild type reduced protein, there's equal probability of going through either of the roughly equal probability of going through either of the pathways. It goes both ways. But whenever you're in oxidizing conditions, suddenly this pathway shuts off, and almost all the excitations go through this indirect pathway 4-2 to 1. And so what we're postulating is that this is you're steering the excitations as a photoprotective mechanism. Whenever you go through this indirect pathway, you have a higher probability of interacting with quenching residues. So the quenching residues I'm talking about are these cysteine residues here, which again are thiol radicals in oxidizing conditions. And also you have this thing called a tryptophan tyrosine chain located near the bottom of the complex. Not much is known about these things. These are chains of tryptophan and tyrosine residues, which are redox active, and they're extremely evolutionarily conserved across a ton of different organisms. They were first found and talked about by Harry Gray at Caltech. They've been postulated to undergo some sort of charge transport mechanism in order to quench those excitations, probably maybe hole hopping mechanism or something. So yeah, again, major result number one, whenever you're in oxidizing conditions, the excitations are steered so that they're more likely to be quenched, which again protects the organism from generating reactive oxygen species. Yeah. So I'm going to move on to the next part. Are there any questions right now?
I actually do have one, Jake.
Yeah.
In your mutated systems, what is being oxidized or reduced because you no longer have the thiols?
That's a good question. I think I don't know. So it's kind of not clear. We're not really sure about that, but it's clear that something is happening, because whenever you look at the ultrafast dynamics of the double mutant, they're actually different, which means that something else is happening in there. We don't really know what the other residues are that are being oxidized or reduced, or something else is going on in there too. It's a good question. We don't know.
As a quick follow-up, these are all heme iron centers that you're showing.
These chlorophyll molecules?
That's correct.
I believe they're magnesium centers.
Okay, gotcha. Thanks.
Yeah, but yes. Okay, I will double check that. So okay, result number one. Let's go on to the next thing. So we have this mechanism of steering the excitations. What we want to do is understand microscopically what is actually happening to the system in order to cause that to happen. So quite literally, what are the cysteines doing to the molecules? We turn to red field theory, which is an old theory that describes the energy transfer rate of the system. And for the sort of experts in the room, it's what the red field theory, you start by treating the system bath coupling perturbatively, which works for us. So what we want to do is understand what the cysteine residues are doing whenever they're being oxidized, reduced, or mutated. And so if you look here at the structure, C49 is sort of between sites 2 and 3. He's like 10 angstroms from both of them. C353 is 10 angstroms from sites 3 and 4. And so what we're saying is that the cysteine residues can change these site energies, and they can change the system bath couplings slightly of these sites. Yeah, let me take a drink. Yes. So what we do is we take the Hamiltonian of FMO, which is pretty well published in the literature. The Hamiltonian is just an 8x8 matrix which shows the site energies of these valence electrons and their couplings to one another. And what we do is we change the site energies, and then we observe, and then we diagonalize the matrix and see what happens to the excitation energies as a result of changing the site energies. We can also, that also will have an effect on the system bath coupling. So right here, this is an important concept. This is something called a spectral density. This is a sort of mathematical object within red field theory. So what this is, on the left here, ignore all this. This is all just sort of amplitude. What this is meant to depict is the relative amplitude of available bath vibrations, which are able to couple the states in your system. Okay, so let me say that again. On the y-axis is amplitude, on the x-axis is frequency. And so, for example, if you look at this little point at 50 wave numbers, this is telling you that there's this many bath oscillations which are able to couple some energy gap that exists at 50 wave numbers, okay? So there's this big hump over here on the left, which is meant to show all the low frequency bath vibrations that are, driving all of this energy transfer. And what we've added in is an intramolecular vibration of bacterial chlorophyll here centered at like 270 wave numbers or something like that. I'll show you why in a minute, why this is necessary to reproduce our empirical data. Really quick, there's also this thing called the Huang-Reis factor, which effectively in our study is just the amplitude of the spectral density. If you lower the Huang-Reis factor, then this whole thing gets cut down a little bit. So yeah, that's the idea. We're changing the site energies and we're changing the Huang-Reis factors to understand what the cysteine residues are actually doing to steer the energy transfer dynamics. And so here's an example of one of these little maps that we calculated. So this is meant to show, this is the tau 41 time constant. The y-axis is the change in site 3 energy, and then the x-axis is the relative Huang-Reis factor of site 3. So what this means is if you start here in the middle, which is wild type reduced FMO, it goes at a certain speed. The energy transfer of tau 41 is at a certain speed. If, for example, you raise the site 3 energy, it speeds up for a little while. If you keep raising it, it starts to slow down. Same thing happens whenever you lower it. Whenever you lower the site 3 energy, it starts to slow down. I hope does that make sense? Yes, Christopher.
Sorry, I was muted. I just have a quick question. Why is there only one Huang-Reis parameter? Shouldn't there be one for every normal mode?
Not in this. That's a good question.
Because usually there's like one per vibrational coordinate, or one per vibration.
Oh, yes.
And this is like a pretty complicated protein, so I think that there's more than just one frequency.
Yeah. No, you're right. No, you're right. Typically these spectral densities. So people represent spectral densities in two kinds of ways. They either have this like big lump of low frequency vibrations, and they use just one Huang-Reis factor for all of them. But then, yeah, they have different Huang-Reis factors for the different specific modes, which is the other kind of spectral density that people use. For us, we're saying one Huang-Reis factor for everything, which yeah might not be, which might be okay. But yeah, so this is just to say that are there more modes in general?
Is the right way to interpret that then as some kind of average? Is that how you would describe it?
An average of like bath vibrations or something?
Yeah, an average over all the modes, or does it represent maybe the one that it couples to strongest? Or
yeah, I think that's probably right. It's an average over all the modes, because I didn't say this, what we're actually doing is we're studying an inhomogeneously broadened system, or like we're studying actually 50,000 Hamiltonians that are averaged over static disorder. And so yeah, it's the relative Huang-Reis factors you can think of as like an average of the available bath modes or something. Yeah.
Thanks.
Yeah. Thank you, Christopher. Okay. So that's it for this one. This is just one example. Tau 41 change in site 3 parameters. We actually calculated a lot of, we made a bunch of these little red filled maps where you have tau 41, tau 42, tau 2,1 for sites 2, 3, and 4. There's a lot of information here. It's just kind of a cool thing. What I'm trying to show you here is that this red field space is actually 6-dimensional. And so what we're trying to do is place our data onto this red field space. So what I mean here is what could explain why the wild type oxidized protein goes slower? Well, if you start here, maybe you could raise the site 3 energy. So that slows down tau 4.1, but that subsequently speeds up tau 4.2 or tau 2.1. Is that depicted in our data? So it's kind of a puzzle. It was actually kind of fun, but also really frustrating to figure out exactly where everything was. But that's what we did, and I'll show you that on the next page. So these are 3 of those maps that I've shown. Just these are the 3 most sort of exemplary ones. So for example, you have the wild type oxidized here. This explains why it slows down. Here's the single mutants, and here's the double mutant. Okay. So what we did, right. So we did this for all 8 of the FMO samples for all 3 time constants that we extracted from the data. So it's a bit of an overfit system, and so the way that we made sure that this was right is that we made sure that the data were that the mappings were internally consistent. By what I mean there is, if you take the wild type oxidized, if you single mutant one, if you single mutant the other, the double mutant should be the sort of vectorial addition of the 2 single mutants, right? So that's true for all of the red field maps, that there's that sort of vectorial addition. Same thing is true for wild type reduced, or for the reduced samples. So that's really a constraint on the system that allows us to show exactly what's happening. So it's actually really necessary for us to study these mutants, it turns out, because they allow us to constrain this data like this. Really quickly, before I go into the next slide, remember the single mutants were faster than the double mutant in the oxidizing conditions. The reason why that happens is you're actually moving through this red region, which represents coupling to the vibrational mode. So when you mutate one of them singly, it gets faster, but whenever you do both of them, it sort of overshoots this vibrational mode and gets slower. So it turns out the data are actually cooperative, they're actually consistent, but it just didn't seem like it at the outset based on the sort of phenomenological time constants, which is kind of neat. Okay, so here's big result number 2. What we see is redox-dependent vibronic coupling is a steering mechanism, okay? So what we're saying is that the FMO complex has evolved a way to steer its excitons by turning on and off the vibronic coupling in its chromophores, okay? So I've shown here the same spectral density from before. This, so like I said, we had to add this intramolecular vibration in order to reproduce the wild type reduced time constant. And this is the statistical distribution of exciton 4-1 energy gaps, okay? So what this means is this mode is able to couple excitons 4 and 1 this much in this entire ensemble, okay? This is the reducing conditions, but whenever you're in oxidizing conditions, this the site energies change so that the 4-1 energy gap is actually detuned from that vibrational mode, okay? So you can see here with these little parabolas that the energy gap is this much, which matches the vibrational mode of the excited state. And in oxidizing conditions, it suddenly doesn't match anymore. And so you get into this sort of like dead region of the spectral density, and your energy transfer slows down. Okay, so that's the deal. That's the big result. It's actually really fascinating that you see this redox-dependent steering, or this is kind of a cool mechanism, because all that's happening is that the cysteine residues are gaining and losing a proton, and they're able to actually modulate the vibronic coupling and steer these excitations. So this is the sort of synthesis slide where everything is put together. Again, we have this steering in oxidizing conditions, or in reducing conditions, you go down both pathways. In oxidizing conditions, you go down the indirect pathway to increase your likelihood of quenching. And the mechanism by which that happens is this redox-dependent vibronic coupling mechanism. And so we can see the connection here, how the organism can turn on and off its vibronic coupling as an oxidative photoprotective mechanism. In other words, it's able to modulate this really quantum mechanical effect in order to produce sort of larger changes in the physiological process of the organism. It's able to actually respond to dangerous environments by turning off its vibronic coupling, which is a sort of a novel kind of evolutionary mechanism that we've found in these kinds of organisms. So that's everything. But of course, whenever you get a result, it spawns 47 other questions. And here's a few of the questions from these results. Number one, how does the redox environment affect other more complicated photosynthetic systems? Like I said, there are photosynthetic systems which are extremely complicated that have tons of chromophores, and a lot of them have cysteine residues and other redox active residues that are really close to them. So maybe redox dependence is a thing in other organisms. Is this vibronic tuning mechanism universal? The mechanism is actually quite simple, which suggests that it's not that hard to evolve. So where else is it in evolution? What other biological functions does it actually stand to promote? And then finally, sort of at a fundamental level, what does this say about how much we understand about vibronic states, sort of, as a fundamental chemistry question? So I want to leave you with this sort of fundamental question here about vibronic states. So, typically chemists think about these states, you can either be purely vibrational state like this, you can have coupling between just vibrational states, or you can have coupling between just electronic states. You think of it as kind of a binary where it's one or the other. But vibronic states really show you that they span the region between electronic and vibrational states. So there's actually a spectrum of different kinds of states that you can be. On the left, I've shown these vibrational states. On the right, I've shown what I would say purely electronic states, but they're actually limiting cases where in the middle you have, once again, you see an excited vibrational state on one excitonic surface coupling with a non-excited vibrational state on another excitonic surface. They couple together to produce a new basis, these black lines, which are vibronic states. And so these vibronic states are really hard to sort of visualize or conceptualize. And the reason is because, they're non-adiabatic. The Born-Oppenheimer approximation was first formulated in 1927. It's completely influenced the way that chemists, it's really hard for us to think about what these states actually look like. Okay, so you have through space coupling of the nuclei, so you have delocalized nuclei, and you also have delocalized electronic states, and they're dynamically interacting with each other. It's really kind of interesting to think about what a vibronic state actually looks like, because they break a lot of our intuition that we've spent years developing. And I should also say that there's many different ways to be vibronic. Vibronic is really kind of an umbrella term. You can be over here on the left and be mostly vibrational. You can be mostly localized onto one electronic surface, but have a little bit of electronic state, or you can be on the other end like this. So I think in the past 10 years or so, people have in my community, the photosynthetic light harvesting field, have really started to try to understand these vibronic states, and have really, like this study, along with a few others in the last few years, we're finally starting to see some experimental evidence for these vibronic states and trying to understand what they mean and what they do. So, it's really cool to be, here at this time, whenever we're just sort of cracking this case open. I'm sure theorists have been thinking about this for a long time, but, experimentally, we're finally, cracking this open. And it's really cool to see how biology has, actually evolved to manipulate these kinds of different states. For the last 100 years, while we're all in our little binary Born-Oppenheimer picture, the whole time evolution has been using, vibronic states, which is kind of interesting. So yeah, with that, I'd like to go through a few acknowledgements, and then I will take questions. Like I said earlier, I'll turn off the recording for questions because I'm hoping that this can be kind of a little bit of intellectually vulnerable kind of conversation. Anybody feel free to ask any kinds of questions, and you won't be permanently recorded. So I have a few acknowledgements first. One, I would like to acknowledge Grit. Yes, Cheng-Han, I'll get to you at the end after all this. Oh my God, Cheng-Han, it's so good you're here. So first, I would like to acknowledge Grit. So Grit has been extremely instrumental to my PhD. You know, I'm super proud of this organization and having been part of it and having been a co-director of it with some people who I think are in the Zoom chat. I think Grit's doing really good work to, you know, chip away at some of the structural issues that are within science academia. I also want to say that it's been really personally important for me because, you know, in this department, I've experienced a lot of isolation and loneliness, and, you know, specific things that are specific to my own identity as a person from an underrepresented background. And Grit has really established sort of a community for me of people who have similar experiences and who can talk about these things and who kind of center these experiences. So that's been really great. I would say, you know, that's been great on its own terms, and that's the most important thing. But I'd also say I wouldn't have had any of the creativity or sort of self-actualization that it took to come up with some of these things if it weren't for Grit. So definitely it's been huge for my PhD. I'd also like to acknowledge the union, which I'm a part of. Admittedly, I haven't been as involved with GSU. It's our, you know, university-wide graduate union. But, you know, I still fundamentally believe, one, that everybody struggles with graduate school, but that a lot of the struggles are, at least in part, for everyone, due to structural issues at the university, at the divisional, and at the department levels. And what a union does, it allows us a mechanism by which we can collectively bargain. All of us in the Zoom chat sort of collectively have the power to make change at the university level. And so, yeah, I just want to plug that here. And I've also met some really cool people at GSU too, and it's been really helpful for my PhD as well. And I want to say, if anybody here wants to be a faculty member, or if faculty members are listening to this, if you're a faculty member that thinks that students should be able to advocate for themselves, then you should support unions too, because unions are one of the best mechanisms by which students can advocate for themselves. So I would also like to acknowledge my PI, Professor Greg Engel, over here on the left. Greg's been really supportive through my PhD for a lot of reasons and a lot of things. Here's some other folks in my group. Here's Lawson, who's in my cohort, and Elizabeth, who's also in my cohort. We've been through this for the last 5 years, which is wild to think about. There's also Chidera and Anna, who are the undergrads I worked with again. There's Professor Sarah Massey, Marco Elodi, Sarah Sohail, John Otto, and Po Che Ting, who have all been involved with this project. Also, everybody else in the INGLA group, we have a good, wonderful, dynamical group with a lot of energy, and all the first years here who are in the group too. And then the Blankenship Lab, which, you know, they were the folks that did the foundational study for this, and they, you know, collaborated with us to send them the samples, or to send us the samples, which we studied spectroscopically. So with that, thank you very much. I will stop the recording and take questions. Hold on.
So